Welcome back, everyone, to the Deep Dive. Today, we're heading into the fascinating world of forensic science, but with a twist. We're diving deep into the realm of proteins, those tiny building blocks of life, to see how they're cracking cases wide open. You see, DNA can tell us who was at a crime scene, but proteins, well, they can reveal so much more. They can tell us what happened and even when it happened. It's like having a microscopic detective on the case. So how do scientists actually unlock these secrets hidden within proteins? What are the techniques they use? And just how powerful is this technology? Joining us today is an expert in the field of forensic proteomics. Welcome to the Deep Dive. So glad to have you here. Maybe we could start with the basics. Can you break down for us what proteins actually are? Why are they so important in forensics?
Imagine a bustling city with workers carrying out all sorts of essential jobs. You've got construction workers building and repairing structures, transport systems moving things around, and even a defense force protecting the city from invaders. Well, proteins are like those workers, but in the city of our cells. They build and repair tissues, transport molecules, and even defend against invaders like viruses and bacteria. And each protein has a unique structure, like a fingerprint, that determines its specific job.
Okay, so proteins are the workers keeping everything running smoothly, but what about proteomics? Is that like the city planning department mapping out all those different jobs?
You could say that. Proteomics is the study of all the proteins in a given sample. We look at their interactions, how they change over time, and what that tells us about the bigger picture. It's like studying the entire workforce of a cell to understand how it functions and what might have gone wrong.
So it's like having a massive database of all the possible workers and their jobs. But how do scientists actually analyze these proteins? I've heard of mass spectrometry, but isn't that a pretty complex process?
It might sound intimidating, but it's an incredibly powerful tool. Think of mass spectrometry as a super-sensitive scale that measures the mass of molecules. For a long time, scientists could only use it to analyze small molecules. But with breakthroughs in techniques like soft ionization, they found ways to analyze big, delicate molecules, like proteins, without breaking them.
Wow, that must have been a game changer. So if we think about a bloodstain at a crime scene, how do scientists use this technology to get protein information out of something like that?
It's a bit like taking apart a complex machine to understand how it works. We call it the bottom-up approach.
Okay, walk me through it.
First, we have to isolate the proteins from the evidence, like your bloodstain, and purify them. Then we use an enzyme, often trypsin, to break those proteins into smaller pieces called peptides. It's like cutting a sentence into individual words.
So they're creating a protein dictionary of sorts.
Exactly. Then we separate these peptide words using a technique called liquid chromatography. It's like separating different colored candies on a conveyor belt. Finally, we feed those separated peptides into the mass spectrometer. It measures their mass and creates a unique fingerprint for each protein.
So it's like each protein has its own barcode at a molecular level.
Precisely. And once we have those barcodes, we can start identifying what proteins are present in the sample and what they can tell us about the crime.
This is already so fascinating. I'm starting to see how this could be used to solve some real-life mysteries, but what happens next? How do they analyze this data and actually find answers? So we've got these protein fingerprints, these barcodes, but I'm still a little fuzzy on how they actually use this information to, solve a crime.
That's where it gets really exciting. We're talking about using these protein fingerprints to answer questions that traditional forensics just couldn't.
Okay, you've got my attention. Give us an example. A real-world case.
Let's talk about forensic serology. That's the analysis of body fluids, blood, semen, saliva, the stuff you find at a lot of crime scenes.
Right. And I remember you mentioned that earlier. But how's proteomics changing the game in this area?
Well, the traditional tests, they can be a bit unreliable. Some of them are what we call presumptive tests. They might give you an indication that a substance is present, but they're not always right. Kind of like a first guess that might be off the mark.
So like a false positive.
Yeah, exactly. And other tests, they're not very specific. They can't always tell you exactly what kind of fluid you're dealing with, especially if it's a mixed sample.
So you need something more precise.
Exactly. And that's where proteomics comes in.
Yeah.
Because every body fluid, it has its own unique protein profile.
Yeah.
And using these techniques, we can identify them with a much higher degree of certainty, even in complex mixtures.
So it's much more reliable. And I imagine that's a huge advantage in cases like, say, sexual assault, where identifying the presence of specific fluids is critical.
Absolutely. It can really make or break a case.
Wow. So we've got serology. What other areas of forensics are being, revolutionized by proteomics?
Another big one is forensic toxicology.
Okay, toxicology, that's the study of drugs and poisons, right? But how does analyzing proteins, how did that help with toxicology?
Well, it's not just about detecting a drug or poison. It's about understanding how it affected the victim. Remember, we talked about those proteins being like the workers in our cells. Well, certain drugs and poisons can actually mess with those workers, disrupt their function.
So you're saying proteomics can show us the damage a drug or poison has done, but at a microscopic level?
Precisely. And one really interesting application is in the detection of synthetic insulins.
Insulin. So we're talking about diabetes here.
Yeah. People with diabetes rely on these synthetic versions of insulin to regulate their blood sugar. Now, in a suspicious death investigation, if someone's found with synthetic insulin in their system, we need to know, was it an accidental overdose, suicide, or maybe something more sinister?
So being able to identify that specific type of insulin is crucial.
It can be the key to figuring out what really happened.
That's incredible. The level of detail you can get with this technology, it's really remarkable.
It is. And it's not just limited to insulins either.
Yeah.
Scientists are using proteomics to identify protein toxins as well. Things like ricin.
Ricin, that's highly poisonous, right? I remember hearing about it being used in, like, bioterrorism cases.
Exactly. It's derived from castor beans, and it's incredibly dangerous. So being able to quickly and accurately identify ricin in a sample, that's critical for public safety.
Absolutely. So we've got serology, toxicology. What other areas of forensics are being, I don't know, I guess revolutionized is the word, by proteomics?
Well, one of the most fascinating applications, and it really highlights the power of this technology, is in human identification. And we're talking about cases where DNA is degraded or unavailable.
Wait, really? You can identify someone without using their DNA? How's that even possible?
It sounds crazy, right? But it all comes down to analyzing something called genetically variant peptides, or GVPs for short. Basically, these are tiny variations in proteins that are caused by subtle differences in our DNA.
So everyone has their own unique set of GVPs.
Precisely. It's like a fingerprint, but at a molecular level. And by analyzing these GVPs, scientists can create a unique protein profile for an individual.
And you're saying that these protein profiles can be used to identify someone even if there's no usable DNA.
That's the idea. It's particularly promising for cases involving hair shafts, bones, teeth, things where DNA might be degraded or difficult to extract.
Imagine being able to identify someone from, like, a tiny fragment of bone that's been exposed to the elements for years. That could be huge for cold cases.
Absolutely. It's a game changer. And it's not just about identifying individuals. Proteomics is also being used to estimate the post-mortem interval, or PMI.
Right, PMI. Figuring out how long someone has been dead. And I know investigators use things like body temperature, rigor mortis, but those methods aren't always reliable, are they?
You're right. They become less accurate as time goes on. And that's why this application of proteomics is so exciting. Researchers are finding protein biomarkers that change in predictable ways after death.
So by analyzing these biomarkers, they can potentially estimate PMI with much greater accuracy.
Exactly. And it could be a huge help, especially in those more complex cases.
This is amazing. I mean, I came into this deep dive knowing very little about proteomics, but this is just mind-blowing. The possibilities are incredible.
It is pretty amazing, isn't it? And honestly, we're really just scratching the surface of what's possible. As research continues, I think we can expect even more groundbreaking applications in the years to come.
So what do you think the future holds for forensic proteomics? I mean, what kind of impact do you see it having on the way we solve crimes? It really feels like we're on the verge of a forensic revolution. But, I'm curious, what are some of the challenges that are still out there? What's holding proteomics back from being even more widely used?
Well, like any emerging field, forensic proteomics definitely has its hurdles. And one of the big ones is the need for larger and more comprehensive protein databases. Remember we talked about database searching, how we match those protein fingerprints to a library of known proteins?
Yeah, the bigger the library, the better the chances of finding a match, right?
Exactly. The accuracy of those searches, it really hinges on having a robust database to work with. And because there's so much variation in the human proteome, because of our genes, our age, lifestyle, all of that, we need those databases to be as diverse as possible to ensure accurate identification.
So it's really like trying to find a specific book in a library. The more books you have, the easier it is to find the one you need.
Perfect analogy. And it's not just about size, it's also about the quality of the data. We need to make sure that the information in those databases is accurate it's reliable, and it's standardized.
And I imagine standardizing procedures across different labs is important too, right? If everyone's doing things a little differently, wouldn't that make it hard to compare results?
Oh, absolutely. For forensic proteomics to be widely accepted in court, to be used as evidence, we have to make sure the methods are reliable and that the results can be reproduced. It has to meet those really high standards of scientific rigor,
Wow.
the kind that are required for legal proceedings.
It sounds like there's a lot of work still to be done, but also, incredible potential for this technology to really revolutionize forensic science.
I completely agree. And, this kind of brings up another interesting question. Do you think proteomics will eventually, I don't know, maybe even replace DNA analysis in some areas of forensics? Or will they end up working together, each one complementing the other to get that full picture of the crime?
That is a fascinating question. It's clear that proteomics can tell us so much that DNA can't, like those insights into what happened, when it happened. But DNA, it still has its strengths, especially when it comes to identifying individuals.
True, DNA is still the gold standard for identifying someone, especially when you have a good, clear sample. But what's so exciting about proteomics is that it can fill in the gaps where DNA falls short. Like in those cases with degraded samples, or when you need more than just an ID.
So maybe the future is really about combining those approaches, having the DNA and the proteins working together to tell the whole story.
I think that's definitely the most likely scenario, and it's the most powerful. You get the who from the DNA and then the what and the when from the proteins. That gives investigators an incredibly powerful toolkit.
It's like having two detectives working on the case, each one with their own special skills, their own perspective, and together they can build a much stronger case and hopefully bring a greater sense of justice.
Exactly. That's what makes this field so exciting. It's not just new technology, it's new ways of thinking, new ways of uncovering the truth. It's about making our world a safer place.
Well, listeners, that brings us to the end of our deep dive into the fascinating world of forensic proteomics. We've explored the science, the techniques, the challenges, and the immense potential of this rapidly evolving field. And I don't know about you, but I'm definitely walking away with a whole new appreciation for those tiny molecules and the incredible scientists who are using them to, really make a difference in the world.
It's been a pleasure joining you today.
And until next time, keep those minds curious, and remember, there's a whole world of fascinating knowledge waiting to be discovered right here on The Deep Dive.